3 with us today. Good morning or afternoon, depending on where you are. There's a good chance it is. Yeah, sure. Evening. Let's go. All right. So we are live on Facebook, YouTube. Great. All right. So let's get started. So today's structure is really, welcome to day 2, part 1, assembly, for everybody who's joining remotely in the future. So today is really structured in a way where we try and do as much hands-on as we can, but obviously with large data sets, sometimes that's not possible. So the flow of today is really, we have a kind of a slow first session about assembly, where we get to talk about it more abstractly. We get to look at some of the programs and some of the parameters, get a little bit of an assessment of how different assemblers work on one data set, but there's not so much hands-on, so we take it a little slow. The second session this morning is about binning, which has a ton of hands-on stuff. Well, you'll be like, oh my God, there's so much hands-on stuff. And we'll be like, cool, that's exciting. So if we run a little bit longer past noon, that'll be okay. We'll just move lunch accordingly to make sure at least we get an hour. And if we need a little bit more time, we need a little bit more time. And then this afternoon, again, we have to slow down because then we talk about annotation. And so while specific examples are possible and we can talk about different options and how you might want to work with some of the data yourself, again, it's one of those things where we can only do so much with a small data set. And then we finish off the day with a phylogenetics assessment, which is also really intensive hands-on. So we kind of go light and then hands-on, and then we go light and then we go hands-on. So if you're feeling like there's not a lot of hands-on happening in the session that we're in currently, there's a lot in the next session. So, but with that, we're going to talk a little bit about assembly. And so the way this talk is set up is that yesterday we kind of got really into like technical stuff. I think at the end of the day, we don't even start the day talking about why we're here in metagenomics and what the real goal is. We kind of only talk about that late in the afternoon. Everything else up until that point is kind of like, well, you're going to get sequence data because that's why you're here. So let's talk about that. So I'll spend a little bit of time walking through, especially from a marine perspective, why metagenomics is an important aspect. And then we'll get into okay, so we've done some metagenomics, we've taken some samples, we have some sequences that we've now quality controlled. What are we going to do with them? What's our first step with that? So that's how it's going to work. Let's see, I can make this a presentation. It's going to be a little while. Okay, so the big question of people who maybe don't think about this, and I'm sure that most of you do, is why are we studying organisms, microorganisms in the environment? And I think a lot of us kind of inherently know the answer to that if we haven't thought about it. Some of us have probably heard terms thrown around before. But really what we want to be doing is taking some type of snapshot that allows us to figure out who's there, what they're doing, and how they're doing it. And so, okay, well, that's great. So why don't we just do something that isn't metagenomics, right? Lots of things live and grow. Microorganisms live and grow in lots of different conditions. Let's just do something like that. And so the first big drawback to that is that the marine environment has a lot of cells in it. So we're talking about on a scale of 10 to the 28th, the aquatic habitats here, the ocean, have somewhere on the order of like an estimate of 6 times 10 to the 28th. So that's a lot of cells to deal with, right? And so even if we were really good at growing things, we probably wouldn't be really good at growing lots, and if there's a lot of diversity, lots and lots and lots of things, because there's a lot of things out there. And then we run into this problem where organisms in the environment really don't like the lab. And so this is a term that's been around for since the 80s. It's called the Great Plate Count Anomaly. And so this has been a common theme in especially aquatic microbiology of, listen, when we take seawater and we put it on a really rich media and grow it in the lab, we see 20 organisms. That's probably what's in the ocean, except that when we look under the microscope it doesn't look like that at all. And so you end up with this complete disconnect between organisms that we can grow in lab conditions and what it looks like is under a microscope. And there's been lots of theories about why this is the case. When this was first being pitched around, and the idea was like, well, maybe they're all just dead cells. Maybe we can't grow them because they're all just dead in the ocean, right? Maybe there is only 20 organisms that grow, all the rest of them are dead. That turned out not to be correct. And so now we know pretty solidly that there's lots of diversity in the ocean, and a lot of it we can't get at currently with the methods that we have in the lab. There are lots of labs out there now who are making lots of progress with being able to culture organisms that were recalcitrant culturing before, but it's still, even if they get really, really good at it, there's still 10 to the 28 cells in the ocean that we have to maybe look at to understand a little bit better what's happening in the nature. So we need this idea of something that we can do where we are not cultivating organisms, and we can do research independently of that. The other thing that we know from this assessment of the Great Plate Count anomaly is that the isolates that we do find do not equal what's happening in the community, in the environment. And so there's lots of, we now know there's lots of crosstalk between organisms. Organisms generally have some type of interaction, either antagonistic or supportive of other organisms in the environment. And so even when we get cultures of organisms from the ocean or other environments, they don't tend to behave the exact same way as they would if they were actually in a natural sample with other organisms that they are used to seeing or not used to seeing. And so using omic methods, and in this particular case, like we're talking about is metagenomics, this allows us to get some information on a broad scale about total metabolism, total interactions, total processes of a whole community. And so what we're really asking for is, how are these organisms interacting and responding to their environment? And so our goal is to try and get genomes from these organisms from this technique, something that we touched on a little bit yesterday at the end of the day. And really the reason for that is genomes are this kind of snapshot of potential that organisms, what they can do, right? So if we have a genome, even if we don't see them doing it, we know they have the potential for doing it. And there's lots of cases like that where we can see genes in a genome, and in the lab, we can't get them to express it. And that's fine. But we do know that there's probably some time where those genes are important, because bacteria are really good at dropping genes that they don't need anymore. And so we need to be cognizant of the idea that a genome is a blueprint. It gives us some idea of potential, and then it allows us to do something along the lines of asking post-genomic questions of, okay, so now we have more information. Can we do something different that we haven't been doing before that might allow us to learn more about this organism in a different context? We're missing some figures here. That's fun. This is just the conversion of the PowerPoint to the Google Drive version of PowerPoint. How exciting. But so in a very broad sense, a metagenomic workflow looks something like this, where we take an environmental sample. We're going to extract some biomolecule. And so we're talking about DNA because we're talking about metagenomics, but you can also extract RNA, proteins. You do something that we talked about a little bit yesterday about this library prep step, shearing, size selection, that type of idea. We're going to go through some high throughput sequencing. This arrow here hides a lot of the stuff that we did yesterday with the quality control and things like that. And we're going to talk about in this step here. No, okay. We're going to talk about in this step here about assembly, and we're going to talk about binning. And then once we have genomes, we can do lots of things on the other side where, we're talking about annotation, phylogenetics. We're going to learn about, we can figure out ways of figuring out what the organism is doing, what their potential is, as well as some idea of where they fall on the tree of life. So we're going to get to all of that today. All right. So right now, the real standard for metagenomics is short reads. And I say right now because if you're aware at all a little bit about the technology and the way it's moving, short reads may be a thing of the past in a not too distant future. We have lots of long read technologies out there that are becoming more and more successful. There are plenty of people to follow on Twitter if you want to have your pulse or finger on the pulse of what's happening with long read sequencing technologies. The great thing will be is that at some point in the future, long read sequencing technologies hopefully will allow us to not have to worry about the short reads anymore. So you might be learning something that will become defunct in 10 years time from now, which is fine. It's a long time. You know, people learned how to do Sanger sequencing 15 years ago, and they still have jobs, so don't worry about it.
There's a lot of combining those techniques.
Yes, right now. So right now there's a lot of combining the short reads with the long reads because the long reads have a lower accuracy level. But if we get better at that, then hopefully short reads go away. It's much easier to work with long reads than it is to work with short reads.
A record nanopore read is 2.2 gigabits for a single read.
Okay, so
as of like a few weeks. What's the error rate on that? It doesn't matter. You lay aluminum on top of it.
Yeah. So yeah, so we're going to talk about short reads, but know that this is maybe just a moment in time in terms of technology and what we're doing. So the question here posed on the slide is, how do we do something like piece those red fragments, which all come from the same organism, how do we piece them together? It's an interesting conundrum. And so one of the things that we can do to try and get at be more successful in this step, and people have done this for some time now, is try and find a sample or an environment where, if we're looking for the red organism here, where they are the most abundant organism. It makes it a little bit easier, especially if we can get very large numbers of a single, or a couple of closely related strains into a sample. We can start to treat the sample a little bit more like it's almost a single assembly of one organism. And so, but there's a really big difference between a situation where it's the dominant species in the sample, where maybe you can get lots and lots of coverage of one organism. And then, as soon as you flip it on its head and say, okay, well, we got the red organism from this environment, but now we think the yellow one's really important. The yellow one's at a much lower abundance level, and it becomes a lot harder to tease apart that signal from the rest of the signal that's mostly the red organism here. So abundant genomes tend to be easier to extract and work with, especially the more clonal the population in a system it is, than the rare organisms. And so you can get something that looks like this. So this is just kind of a mock of what a community might look like. And so you have this setup first, community 1, where you have a really abundant organism. This 100 is not percentage, because obviously that adds up to more. It's just a generic 100 here. So this first organism is really abundant. It's more than half of the sample. In a sample situation like that, it will potentially be easier to find that organism signal out of everybody else's signal and do something specifically about pulling that one genome out. Compare that to sample 2, where everything's a little bit more even. That signal in one sample is kind of lost, right? So if you have a lot of short reads, even if you get, like we talk about in a second, some assemblies from it, it's really hard to tease out what one set of reads or contigs belong to one organism, especially on the short scale, because things like the inherent properties of the DNA, like tetranucleotide frequencies or GC content, don't exist at such short levels of like 100 base pairs. You just don't have enough length to get a signal like that. And so you end up in the situation where, despite having some, equally abundant organisms at some maybe high copy number, because they're all in the same sample at the same depth of coverage, it's really hard to pull out any one organism from that. So if you have some way of estimating how diverse your community is, maybe like using amplicon sequencing, like we talked about yesterday with Chime 2, you'd be much better suited to be trying to figure out what your sequencing demand is. And so to go at the very extreme of this example is, so this might be something like a soil environment here, where it's not just a handful of organisms like in sample 2 here, which maybe are still feasible, but you get into this really complex system where even the most abundant organisms are a fraction of the total community, and then you have this really long tail of organisms. And in the marine environment, we know that there can be organisms that are fairly limited in abundance and still have a really large impact on the system due to their activity levels or the key process that they're functioning in. And so when you're thinking about complexity and where we need to go in terms of sequencing demand, we can think of it kind of on this kind of like plateauing system that has some range of information. And so, with sequencing demand of organisms that are isolated is fairly low. Short reads still require some couple million sequences, but I did see an ad the other day. So for 2 million sequences, you can pay $100 now. That's about enough if you have an isolate to recreate a whole microbial genome at that point. So $100 doesn't sound too bad. And then as you get higher in complexity in terms of the communities that we expect to find there, your sequencing demand increases. So the next step would be microcosms or enrichments, where you can have a couple organisms working together in a limited environment and some maybe differential difference in abundance. You get to simple environments like the acid mine drainage, which we talked about yesterday, is our example of the first successful isolated genomes from metagenomic work. And so these are real simple, natural communities. They only have a couple members to them. Hypersaline environments, which are still extreme environments, which have limited amount of diversity. The human gut microbiome, where right now it's estimated that we can maybe culture about 80% of the organisms in the human gut. And so that makes it a little bit simpler to work with. And then we get to planktonic marine environments and soil environments being the most complex, where there's a lot of diversity. And that requires a lot of sequencing to get to. So there have been lots of studies previously about what to do if you can't get good assemblies. And so this is something that we touched on yesterday about the idea of low read depth versus high read depth metagenomes. And so even if you go through all this effort and you get a metagenome and you can't, and you go through all of the steps and you call everybody you know and you do all sorts of tweaks and adjustments and you try to make the best out of a situation that doesn't seem to be working, there are lots of things that you still can do with your reads that you're getting from your metagenome, even before you get to the assembly standpoint. And so this is an example from the Global Ocean Survey, where they were taking genomes of organisms that they had already created, and they were taking the reads from their different sample sites, colored here in different colors, and they were mapping them back to those original genomes and seeing where the environmental diversity matched up to the diversity that we knew of either in culture or from a genome that they had already. And so there's lots of ways of doing this. You can construct functional profiles, which we talked about a little bit yesterday, look for genes of interest. We can do these comparisons to samples, and there's lots of ways of doing that, which we're not really going to talk on. So these min-hash and k-mer-based methods, which tend to be faster than things like BLAST. If you have an impulse to take BLAST and take your big giant metagenome and recruit it to a bunch of genomes, see me first. There are other ways to do that a little bit more easily than just a raw BLAST against stuff. And then this example here of fragment recruitment onto reference genomes. So, it's not all bad. We have the ability to do this pretty quickly. It provides a general profile and it has a broad view of composition and function. But then we get into this problem where, we might not have accuracy when it comes to phylogeny. We might not have accuracy when it comes to which genes belong to which organism. And so it's really great that we can tell process A and B are potentially happening in the system, but it would be really cool if we could see if process A and B are actually in the same organism, because there's some, interest that we have in trying to figure out what the limits are and, why these genes either are or are not together in a sample. And as always, this kind of always relies on having a reference genome. And we know that we don't have reference genomes for a lot of organisms in the environment. So this is our metagenome workflow. So this is generally what we're going to mostly cover for something today. So, and have covered yesterday. So we start with our raw reads up here. We've gone through generating high quality reads through quality control process. So we're not going to go directly into this too much yet, fragment recruitment and things of that nature. But now we're going to talk about, this morning, we're going to talk about assembling information here. We're going to talk about annotation and prediction. We're going to talk about binning, and then along the way, talk about how you would be interpreting biological, interpretations of this. And so this on the side here is a very interesting way of thinking about how much data that we are working with and what actually ends up happening and what we end up analyzing on the biological aspect of this. So in this particular example, this figure is suggesting that maybe if we're, got some money and we're able to put it towards our sequencing effort, we start off with about 10 gigabase pairs of Illumina sequencing. So, a lot of sequencing. We saw yesterday that quality control throws out some fraction of that, right? So we can go easily from 10 to 7. And then, unfortunately, sometimes when we do that assembly step, we can go from 7 gigabase pairs to 0.7, right? So the amount of information that actually assembles into something that we can see and has meaning can be a really small fraction, ultimately, of what we got off the sequencer. And then if you keep going further, so now we have some assemblies and we want to turn them into protein coding setups. Now we're only dealing somewhere on the order of 70,000 proteins that we're looking at, right? So we've gone from this very large gigabase pair data set that is very big, but we've needed something that big just to get to the point where we're analyzing something along the lines of 70,000, 700,000 proteins. And so every year, this particular slide becomes more and more out of date. And so these are some methodologies that people have used in the past for their next generation sequencing platforms. It's already, you can see it's out of date. So PacBio is the long read version that's here. Nanopore wasn't even on the scene really so much a couple years ago. So you would add another whole column there. We might pull off ion turn because I don't think anybody does that anymore. The first column here is Sanger sequencing, which is still around. You can still do it. It still is the most expensive per base pair, which is great. But there are, this table breaks down an idea of what these different methodologies from this point in time are good for in terms of throughput, cost, quality, and then if you can use them for this whole genome shotgun approach of metagenomics. Really right now we have the idea that the Illumina sequencer is the one that everybody's using, but in the future, these long reads, like I said, will probably take over. So we're going to talk a little bit about different assembly methods and assemblers. So a lot of the original sequence assembly methods, back when it was Sanger, back when you had longer fragments, back when the data wasn't as large, relied on a type of assembler called an overlap layout consensus assembler, or an OLC. And so these are less common nowadays. And this was really, in the grand scheme of things, a brute force way of doing things. You would take a fragment of DNA and you would compare it to all of your other fragments of DNA, and you'd hope to find an overlap. And if you found an overlap, you could say, oh, they belong together, and you'd stitch them together. And you keep doing that until you generated a longer sequence. And that worked really well when you only had 10,000 sequences, when they were all 800 base pairs long. That can work pretty well. And it will probably the type of assembler that we get back to when we get into a realm where we can start generating 10 KB fragments or 100 KB fragments from these long read systems. So what is old may be new in the future, and what is current may be old. So it'll get real weird and wacky going forward. The other type of assembler, which has really become, really become the standard going forward, are these De Bruijn graph assemblers, which break down your sequence into certain letter size words. So they're called k-mers. In this example, where they're broken into 2-letter words, so they're called tumors. Generally, this works on some other scale of 20 MERS or 22 MERS, and they scale upwards, right? A lot of the assemblers listed here iteratively process through increasingly longer and longer k-MERS to find what essentially comes down to is a map path. So you're able to draw connections based off of the overlap of these k-MERS and create a complex map in digital space that then the computer is able to pull out the most parsimonious path to travel to explain the data. So some examples of overlap and consensus assembly are these assemblers on the left here. So they include the Celera assembler, which is the assembler that assembled the human genome back in 2000. Mira, Nubler, the Amos package has Minimus in it. And so essentially what they're doing here is what I said with the overlap, they're finding areas that overlap between fragments, and then they're stitching them together and creating one longer sequence. Yeah. So when you're doing this with using long fragments, you ending up with gaps, normally due to the fact that you just don't have the actual sequence in your sample, right? So if you're comparing this to what the Sanger method looked like, is because you're only generating 10,000 or 50,000 reads, right? So you're not getting at every organism in there. You're not getting at every fragment of DNA. There's just too many organisms in a complex system to do that with. So if you're missing something, it's generally because it's not there, which is in comparison to the De Bruijn graph assemblers, which is if you're missing something, it's generally because you end up in a situation where you've generated what is known as a bubble, where there are lots of equally likely paths for the sequences to take in terms of creating a full-length strand, and the assembler can't reconcile that, so it'll break at those bubbles. And so you can get interesting things that look like this. So this is two closely related organisms here that have created this really long, huge bubble. You can see it branching out like this. And so that's just because, they have some similarity at both ends of this. And then there's this divergence that happens within their genomes. And the assembler is able to reconcile and say, oh, well, I can see that there are two paths here to take. But generally at something like this, you wouldn't know this point here and this point here would break, and you wouldn't be able to figure out exactly where, what caused that and why it happened. So in a lot of times, De Bruijn graphs, if you have a gap, because you are doing it at such high coverage, the sequencing, if you've planned out correctly, you might actually be in a situation where you have the information, you just can't assemble it because the data is so complex that we don't have a way of pulling it out. And so there's a lot of parameterization that can go into genome assembly with these De Bruijn graph assemblers. And we're going to talk a little bit about them going forward, just to highlight them before we get in there. Minimum length of overlap. And so that's an important level. Like, what do we want to call success? If we make this number too low, we're going to stitch together things that don't really have a true overlap. If we make it too high, it might not be working. It won't work effectively. K-MER size. And so we'll talk about this as we go forward. A lot of the more modern assemblers will give you defaults to work with. And if you go back to 2012, when everybody was still trying to figure out what the way of making it long enough fragments were to actually do metagenomes successfully and pull out individual genomes, there's lots and lots of literature about, well, which k-MERs do we pick? Well, in this sample, we picked this subset, and that worked really well, but that doesn't work in the next sample. And so we had to pick a whole different set of k-MERs. So I'm going to bring them to your attention. So that way, if you run some of the more modern, useful assemblers out there and it doesn't work, you can always do something like play around with the k-MERs to see if we can make it work a little bit better. Yeah.
If you have different metagenomes,
Mm-hmm.
Do you process them individually to parameterize the best way possible for each metagenome, or do you do the same parameterization across all genomes to make them comparable?
Sure. So the question, if anybody didn't hear that, was what happens if you have different metagenomes? Do you parameterize them all the same way, or do you parameterize them individually? The answer to that is it depends. if you are working from the same samples, right? So if they're biological replicates, or you have some type of incubation system, right, where you're monitoring the same kind of organisms over time, having some type of homogeneous parameterization makes sense. If you're comparing completely different samples, you've done different things with them. You're going to compare your soil to your sediment and your sediment to your water column. There might be some argument to be making, like, okay, well, these are different systems. I expect them to have different levels of complexity. Having a different parameterization, shoehorning one parameterization on another sample doesn't make a lot of sense there.
But if you want to compare specific functional attributes in one set of genomes versus another, water column versus sediment, even if you parameterize them differently and process them differently, they will still be comparable or no?
Yes. So the question is if you parameterize something like water column and sediment differently, can you still compare the output? And the answer is yes, right? So the goal here is that you're just trying to come up with the best way to coax the algorithm to work with your data. And the output's always going to be the same in terms of like you're generating contigs that have biological data on them. The goal, and if we're doing this correctly, and we can believe the science behind it all, that biological information won't change overly dramatically. But it's going to be in the same way that you wouldn't expect necessarily the same metabolic functions to be in the sediment and the water column. You might have different ways that those assemblies are going to work. The best way to kind of like come back and solve some of that problems, and we'll talk about this, is to actually use the reads that you have from your metagenome to recruit back to the assemblies that you generate, and you'll get a better idea of how successful you were. And if there's anything like, you would have to find a really big, egregious error at that point for you to be like, oh, well, I can't compare this data at this point. Yeah, that's what I'm going to say on that. Some other parameters that we'll have that can be include things like mismatch penalties, gap penalties. So this is very similar to if you're familiar at all with using BLAST. BLAST gives you a ton of options about what type of penalty scores you want to use. How often do you want to include a gap? That's all part of an assembler too. So you can peruse that information and try and hunt down what might work and might not work for your samples. And ultimately, at the end of the day, what we're hoping for is outputs of contigs and or scaffolds. And so I won't talk too much about what the difference between a contig and a scaffold is. They are technically different things, but for right now, we're just going to assume that they're the same thing. Okay, so this is the hands-on part. And so don't be like, oh, right, we're going to do some assembly now. We're not. It's going to be a lot of like, well, let's look at what this program does and figure out if there's anything we can talk about here. So we're going to move into, if you haven't yet, the subfile called assembly. So we're going to go cd home C-DEBI ecogeo assembly. Take a look what's in there. So a lot of what's in there is actually outputs already, because like I said, you can't do a lot of this on your laptop, and it takes a while to run. So what we're going to actually be doing is we're going to look at the parameterization options for a couple of these tools. So there's a bunch of assemblers installed. The three that we're going to look at are all the Brew and Graph assemblers. And so you can actually type these names in. So you can type in mega hit, you can hit enter, and all of these will generate some information about what they can and can't do. And this is just to give you an idea of the type of information that you might want to think about and the type of information that's already assumed for you when you start working with the data. And so this example in mega hit, you have to be very specific about the type of data you're giving it. So either paired FASTA or FASTQ sequences, or interleaved, as we talked about yesterday, interleaved being the version of, instead of two separate files where each of the reads correspond to each other in order, interleave, you take those two files, you mix them, and then the reads that pair with each other are right on top of each other. So it goes, read one forward, read one reverse. So you have to be very specific about that. You can include multiple metagenomic libraries here. And so you just add those by adding commas and pairing the reads together in the correct way. You can add any of your singleton files that we generated yesterday through the quality control steps that can handle that type of stuff. And then we could get into the arguments. So these are the basic arguments that they allow you to work around with. So these are optimizing your k-mers and your k-mer sizes and picking your min and your max as it runs. You can talk about if you want them to merge bubbles or pop bubbles, things that we've talked about before. And so there's, this is a general outline of the options that you can work with. The MegaHit manual is actually pretty good about giving you details about what each of these actually mean and why you might choose a different parameter than the presets. So there are a bunch of presets that come with a setup here. So this example here that works pretty well for a lot of samples is this meta sensitive, right? And so this parameterizes the assembler in a specific way. It sets the k-mer minimum, it sets the k-mer maximum, how often the k-mers increment and the step that they take when they increment. And so this can work. So this is like always like I wonder what the base parameters are. And so this is always a great way of starting. But you can see that there's lots of other options here, including one that they call meta large, which are they claim for large and complex metagenomes like soils, bulk, which is so standard sequencing and just trying to assemble sequences that you don't know much about. There are ways of setting how much memory your computer uses, how many CPUs and GPUs it uses, where you want the output to go, where you want to store temporary files. All of this information is in here. And that allows you to come back, in the future after we've had today, be like, I don't remember anything about assembly. Right, for the assemblers, a lot of the information is stored in this, in these help statements that you can then be like, oh, okay, so I have to think about these things again, right? So it's not something you have to memorize and walk out of here and be experts on. So if we make it small again, so we can do this again for IDBAUD, and that will give us a different list. And you can see some of the parameters actually have similar names and similar functions, right? So again, your minimum and maximum k-mer can be set here. You can set your step size. You can talk about not merging bubbles, using local assembly values and not coverage. And then we can do this one more time with spades.py. And again, it's going to output a similar message, but different in its own way. And so spades right now is in version 3 and is, at least based off of what's in the virtual machine, I actually don't know off the top of my head what the most current MegaHit version is. But spades the version 3 is so new, even at the time of this, and they've been making more and more adjustments as it goes. They even have options here of combining your Illumina sequences with your nanopore sequences, right? So if you wanted to have a long read, short read combination metagenome going forward, Spades has that built into it already. Something to highlight. Okay. So we're going to work with a little bit in this discussion about this artificial metagenomic data set from this environmental micro paper in 2013, where they took the DNA of 62 organisms from a wide range of gram-negative, gram-positive, archaea organisms. They had these organisms in culture, took their DNA, sheared their DNA, mixed them all together, and then did some Illumina sequencing on these genomes. And so we can use this I use this as a standard for doing our comparisons between the different sequencers. We could say, okay, so we put in the same information of a slightly complex system of 62 organisms that we know of. How well do the assemblers actually assemble back meaningful information that could be mapped back to these original genomes? And so we'll talk about that as we go forward. And so I can make this big again for a little bit. So here's an example of how the sequences were processed. Just like yesterday, using Trim-O-Matic to remove the elements that we didn't want to be part of it. Yeah, and so more information about what black means. And so then I have examples here of just the commands I ran. for the assemblers for this particular data set of 62 organisms. And so, here's megahit. You can see right here that using the defaults makes it fairly straightforward, right? There's only a couple, you guys are all experts. You saw some of the, stuff that we did yesterday with Trim-O-Matic, where it was 10 entries of like, well, we want to make sure we know where the file is for this, and we're going to have these settings. This is basically like, use your preset metasensitive. Here's read 1 in the forward reads. Here's read 2. Please give me an output, right? So it can be pretty straightforward if the presets work for you. For IDBA-UD, you have to take the first step of taking your FASTQ sequences and interleaving them, like we did yesterday with FQ to FA. We also have to convert them from FASTQ to FASTA. And so there is some element of megahit and spades, which actually uses the quality information to make judgments about which, if it comes across a small enough bubble and one of the reads is really poor quality and one of the reads is really high quality, it will make a judgment call. The algorithm will make a decision that will then retain the better read compared to the poor quality read. IDBA-UD doesn't actually do that because it removes the quality scores to simplify its algorithmic search. But again, here's an example of what it looks like. So we input our merged interleaved reads. We ask for an output. We tell it how many CPUs to run on, and then it runs. Right. So again, with the presets, it's not overly complex. It allows you to run these processes. If you have a computer with sufficient capacity, and depending on how really large, if you're doing 100 gigabase pairs of sequencing, this would take some time, and you would maybe hit yourself over it. But if you have a big enough computer, running iterations of this to see which version or which slight modifications to the parameters maybe will only take you a week or 10 days or 2 weeks, right? If you're able to run one a day and kind of look at the output and compare the outputs over time. And so, in the grand scheme of things, that seems like it could be a lot of time spent assembling, but once you have the assemblies and you're happy with the output, you don't have to really come back to them. You get to keep working downstream, and then you have some knowledge about how it worked and why it worked. And then lastly, the example for spades, and using their metagenomic flag for this. Again, so this is again a fairly simple input where you tell it where to put its output. You tell it what parameter to use, which is their meta built-in parameters. You give it read 1, you give it read 2. You tell it how many CPUs to run. And then this is one for spades where if you run into a problem in the future, this might be the case, is you have to set the memory maximum that it can use. So in this case, it's 600 gigabytes of memory. The default is 250. So if you have a complex system and you don't change this number, even if you have a computer that has an infinite amount of memory, you might run into problems because Metaspades will automatically shut it off after it reaches 250, which can be problematic. And I've had people who've had that problem of, I have 1.5 terabytes of memory and it says that my sample's not working, and the sample that's slightly smaller than it works and this one doesn't. What's happening? Oh, have you set the flag for the memory? Oh, it works now. Great. All right, cool. Well, that's awesome. Glad I could help. And so, like I said, so that was all run on the metagenomes for these 62 organisms that were artificially mixed together. There's a fun program called QUAST that allows you to do very broad comparison. So QUAST allowed me to take all of the reference genomes that those organisms came from and put them in a file, and then take the reads or the assembly outputs from each of the assemblers and say, how well did these actually match back to the reference genomes. And so I did that by looking at only contigs of a certain length. So I picked 1,000 KB right here because 1,000 tends to be the very lower limit for what you can work with going forward in terms of assembly, or not assembly, for binning. And that still can be pushing it a little bit. A lot of people will work with only things that are maybe 2.5 KB in length, so 2,500 base pairs or longer. And so, 1,000 is really, at the bare minimum, what we can call like a real, a decent assembly length at that point. And so again, this takes a little bit of time, but you can run cost against the output from all of these, all of the assemblies that we did, and compare it against the reference genomes, and then have some output of understanding how well it worked. And so we'll get to that. There's a nice table at the end where we're going to be like, and here's what the difference between the assemblers were for this process. And so while contigs are great and mapping them back to references are great, there's also lots of other things to consider going forward about what is a successful assembly. So one of them that is brought up frequently is this idea of gene prediction, right? So is your assembly method generating fragments that are long enough that you can actually start to predict proteins off of the information, right? And so, this might make, you might be like, duh, that makes a ton of sense, is that the more genes that you're able to generate from your assembly and look at, the more information you have to work with in terms of a biological interpretation, right? So if you have two assembly outputs from different assemblers, and one has a lot of small fragments and one have a lot of big fragments, and you go and you predict how many proteins you can actually pull out from that information, and one has twice as many, that might be the data set that you want to work with, because that's going to have more data for you to interpret at the long time, as you go forward. And so we'll talk a little bit about examples of annotation here as a little segue to save time in some of the other sessions. And so what we're doing when we're annotating is we're looking for specific features. And so we can do things that are looking for non-coding features and protein coding features, right? So our non-coding features include 16S, 23S, 5S if we're working, our RNAs if we're working with bacteria or archaea, the tRNAs, and then protein coding genes. And then there's lots of these other elements that people will commonly look for in their samples, including RNases, CRISPRs, insertion elements, riboswitches, calculating how many pseudogenes are present in your genome can give you information about rates of evolution and how your organisms are different from other closely related to organisms. There are lots of tools that we can use to do this. Essentially what these tools are designed to do are look for patterns in the sequence, looking for start and stop codons, looking for ribosome binding sites that have been coded into the algorithms, and try and find setups where it can find a binding site, it can find a start codon, it can find a stop codon, and predict a protein. There are lots of ways where this fails, right? So you can imagine situations where you have a fragment of DNA in an assembly in a contig that's short or ends abruptly in the middle of a sequence. And sometimes these prediction tools are actually still really good at that, right? So you can have a situation where it can find a ribosome binding site and a start codon, and the gene runs and it hits the end of the contig. And these programs can be like, okay, well, we got most of it. It doesn't have a stop codon, but we can pull out the beginning part of it. And you can have the reverse happen where if you're on the beginning part of the contig, you might have a stop codon. And if there's a frame that works going upstream where there's a predicted protein that way, it might be able to pull out fragments of your genes that you're working with too. Not necessarily a full start and stop.
How does it do if you've got on the beginning of one contig and the end of another contig chunks that match up with each other?
So the programs that are out nowadays for predicting coding sequences do not work with those. So you end up with, so we can talk about this a little bit, but when you get to the annotation stage, you probably end up with especially if it's a sufficiently long piece of the protein on both sides, you end up with a count of 2 instead of a count of 1 for the one protein. And so there are a bunch of tools to do this with. And so if you're looking for RNA prediction, there are specific tools that use this. The best methods actually take into consideration the secondary structure of rRNA, like Kristen mentioned yesterday. And so there's a couple of them here. These are hot links that will go out to various tools that you can use that are online tools. If you have a sequence that you want to look for, the rRNA sequences. Also for tRNAs, there are tools that work for that. So there are lots of problems with these programs that we know of, but it becomes one of those things of like the devil you know is better than the devil you don't know. And so, you know, our RNAs work really well for ones that we have models for, but it doesn't work really well when we don't have a model. Case in point would be when, you know, the CPRs, the candidate phyla radiation organisms, we discovered that they actually have lots of these like insertion sequences into their 16S. And so until we found an organism and somebody mapped what a 16S looked like for those organisms, we didn't have an effective model to be used. And so if you ran a model on a genome for a CPR and it came back without an rRNA, you'd go, that's weird. I don't know what happened there. And so, you know, we know that these databases are incomplete and it can take some time to figure it out. And then in a protein coding situation, you know, not all organisms have exactly the same way of translating their proteins. You can have all of these interruptions that happen as a result of the assembly process, not necessarily generally because of true biology. So you might have these insertions into your sequence that might have a premature stop codon, and that might not be real. It might be just the impact of the technology that we're using. But at the end of the day, while there are lots of protein calling programs out there, one at this point has kind of really risen to the top of the field. It's called Prodigal. You might be familiar with it. You might not. This might be the first time you're hearing about it. And so this is a pipeline or a program that's used in lots of pipelines already out there. And it will predict your coding sequences for genes that you have. So we're going to do this right now with a subset of the assembly from the SPADES output of the 62 organism subset of genomes. So we shouldn't have moved too far, but we'll make sure that we are in our current directory of home C-DEBI ecogeo assembly. Okay, and so there's some, I'll move this up just a tad. And so we can type this all out together. So Prodigal, which we can tab out, our output, which we're gonna ask for protein translations. Right now, we're gonna call it a temporary file, and there's a reason for this madness that we'll get to in a second. So we'll call it temp1.orf.faa. And so this is just some notation. So we're calling opening reading frames, which is what it's called when you have a start and stop codon, and we're gonna output it as a protein. So in this case, we're using the notation FAA. So this is a FASTA file with amino acids. Ha, easy. Okay. We're going to add another flag with a -D and call temp1 orf. This one has an S. Oh, this one should have an S too. Anybody? .FNA. So now we're going to output not only the predicted protein, but we're going to output the predicted coding sequence, the open reading frame with the start and the stop. So the DNA sequence here. We're going to put in our input, which is SPADES _ SRR 606249.subset.fasta. We're going to use the flag -M to treat missing ends as a stop, or missing sequence, or like a row of ends as a stop codon. We're going to output a table file, which we're also going to call temp1.text. And then we're going to say that the version of this is a metagenome, as opposed to a single organism. It treats it slightly different if it's single organism. And then we don't want it to show all of its output while it's working, so we're going to use -Q. And once we have that, we can hit enter. Yeah.
So what if you're just trying to look for a particular exon within a gene that has, you know where I'm going with this?
No, I don't, but go ahead. The word exon already excites me to a level of...
But there's different frame shifts. Like, so let's just say like what you amplified is a particular exon of a gene. Yeah. Or at least you're flanking the exon. Mm-hmm. How, like, you might, the amplicons that you might be getting off might not be the first nucleotide in that frame. Yeah. Does this deal with trying to find which one, where the frame is for each sequence?
So I have a little bit of bad news,
Yeah.
which is that predicting proteins off of specifically amplicons is not something that this program will do.
Oh, wow.
Right. So it needs that extra information. So if your amplicon region had all of the markers, right? So if you had a ribosomal binding site, start codon, stop codon.
Okay. This is just for ribosomes.
Yeah. It looks for a particular pattern that it needs to see to register it. And so it needs at least some set of those three combinations. And so it might work. It might be worth taking a shot at it if they all have start and stop codons. But if they don't,
it would be like middle exon.
Yeah. So that's going to fail miserably with protocol.
Is there programs out there that do that?
I don't have an answer to that off the top of my head.
That's been something I've been searching for 5 years.
And you haven't written it?
No.
Yeah. So that's a great question. If you've been looking for it for 5 years, there's a good chance that it might be missing in there and out there in the.
It might not be looking in the right place.
Might be looking at the right place. Yeah. So we can talk about that more.
Yeah.
Talk about exons. Yeah. It's not something I think about on a daily basis. I can tell you that. All right. And so the reason why we were calling these files temp files for right now is all held in the secrets of when we look at it. So if we do a less of temp1.orfs.ffa, look at the output, I can move this around just a bit. Or actually, I can move my face instead of the. So what happens is we get the name of the protein that we're working with, right? So this uses the contig name that we provided it, and because it's spades, there's all this extra information about coverage and length. It adds an underscore and then increments up from there. So this is the first protein it predicted. This is the second protein. And then it tells you based off of, so this is all from one contig here. This is all from one contig here. It then actually tells you where on that contig it predicted the protein, right? So this says it's between the second base pair and the 784th base pair. This one then found the next gene starting at 982 going to 11428. There's a whole bunch of information that starts here including is this a partial sequence? Does it have a start and stop? What type of start codon did it have? What type of ribosome binding motif did it have? How much space was there between the ribosome binding site and the first codon of this sequence, and what its GC content is? So there's a lot of information there. But as we know, going forward, that having all that information might not be useful. So what we're going to do on the next slide is do a quick way of adjusting that. But we can ask this question of how many sequences or proteins did we generate? So if we do it this way using our grep method from yesterday with a word count -L, we can hit enter on this. So it'll tell us, so in that moment that we waited and paused, how many proteins did we predict? We predicted 5,603, right? So that's what's in that protein FASTA file right now. And so, I showed you with less, but we can also do this as just a reminder of yesterday. So if we grep the protein file and then use head, pipe it to head like that, we can actually output the first 10 outputs of the header lines and have some examples. So these are all going to be coming probably from the same contig. Yeah, they all do. And they're all, you can see the numbers going up as it increments where it is on the contig, the protein is predicted. Orientation, I forgot to mention this, the 1 is forward, -1 is reverse. So this is, the 7th protein, 8th protein, 9th protein. So this one has both a start and a stop, and it goes on and on like that. And so we have all that information to work with, but we might not want to work with all that information. So there's a quick way of working around this. So we're going to use cut from yesterday. And so this is actually a really nifty way of doing it. So right now, the information on the header line are separated by spaces. So you remember yesterday that cut works by default using tab. So we're going to have to adjust that. So we're going to use the -D flag for changing the delimiter. And then, and this is pretty nifty, because the header line has spaces, we can tell it, pull off just everything that's before the first space. And that will be just our ID, right? So that's all this information that's right here. And then because the sequence is all just one line, it's also the first element in its line, right? So when we do and tell it to cut on just the first field, so if we're looking at it as a table, just that first element of the table, we actually pull out both just the header ID and the sequence information. And so if we do this, if we do cut dash F1 dash D, and then you do open quote space close quote, temp1 orfs, and then put some output here. So in this case, we'll name it after the spades part, call it a subset, and we'll just add the orfs.ffa part to it. Which will run really fast. And then if we look at it, you can see we've gotten rid of all that extra information right now. And so the extra information is useful, but on a grand scheme of things, it might only be useful for a short period of time. And then really what you want to do is just be manipulating the protein sequences that you're working with, or the coding sequences that you're working with just the header and the sequence information. So that's one way of cleaning that up. And we can repeat that for the nucleotide sequences. Just make sure you change that to an FNA at the end and hit enter. And so we can take a look at that. And so again, so this is information. So this is the exact same information, but untranslated, or to be more accurate, the proteins are actually translated from this sequence right here. So you can work with both if you're interested in working with both. And so now if you look, so we have these temp files that we don't need to work with, but now we've taken the starting spades config output that we were working with, and then we generated both coding sequences and then the open reading frames corresponding with those coding sequences. So we touched on this very Herod. It's always fun to be able to do this as you go. Known issues with gene callers include the fragments at the ends of the contigs, right? So you end up with fragments of genes. And as we said, that can lead to situations where you're counting things multiple times. If you don't always, so this could be a real problem, if you don't mask the rRNAs before you do this, or if you're not aware of where the rRNAs are in your sequences that you're giving them, what can happen is that these prediction methods aren't particularly smart, and they can find proteins inside your rRNAs because they're just looking for start and stop codons. And so there's actually a whole, if you go to, it might be solved now on NCBI, but if you, for a long time, you could go and you could translate your rRNAs, find the coding frame that generated a protein, blast that into NCBI, and get genes that came back. We know that rRNAs can't be translated to protein. They're not coding in that way. And so this was incorrectly done. And it kept happening. People would do it all the time. They would get back, in the early days of Sanger sequencing, they would get their 16S back. They would translate it. They'd be like, there's a protein in my rRNA. I'll submit that to GenBank. And they would just keep going and going and going and going. So for a really long time, and it might be solved, I actually haven't checked this in a while, you could have people who were submitting proteins from their rRNAs, which was frowned upon, not great. So you should watch out for that, to be careful. So if you think you've discovered proteins in rRNAs, let me know. I'll tell you right now, you haven't. It's happened before. And then very short genes really actually fail in a lot of with these methods. Both Prodigal and one that I didn't talk too much about, which is Frag Gene Scan. They have a set parameter in there which says a protein really can only exist if it's above a certain length. So if you're working with an organism that generates short proteins, right, which we know happen, they do exist, 30 amino acid proteins, 50 amino acid proteins. If you're working with an organism that has that capacity, these methodologies will miss that. An ever-increasing number of blind spots. Don't worry. We all know they're there. It's okay. And so we can do this as well for the full data set. And if we did that, we can count how much this worked. I think this is going to take a little bit of time, so we don't have to spend too much time doing this, right? Because I subsetted it. Why would we want to do that? I'm going to skip this slide because that might. So right. So if we use this on a subset to get to do it before, and it, it paused for a little bit, it took about a minute. This is the full data set greater than 1,000 contigs. We don't want to really spend time waiting for that. We can just move on to the next thing. Okay, and so questions? Yep.
Okay, before we move on from gene prediction,
yep.
Can protocol handle overlapping genes or overlapping?
No. So the question is, can protocol handle overlapping genes? So like in like a viral situation? No, it will only predict one based off of what I'm aware of. You have to use a specific viral setup to look at that type of situation. Not off the top of my head, but we can talk about it.
Some bacterial genes also.
Yeah, yeah. It actually does a little bit better with the bacterial ones than it does with the viral ones, I think. Because they tend to be much shorter regions as opposed to like whole open reading frames. And so we've talked about assemblies, how we do them. We've ran them. They're waiting in the wings. We're going to talk about what the results look like for a second. We're talking about how we predict proteins so we can compare our assembly outputs. One of the other ways we can do this, as I mentioned, is you can take the quality controlled reads that we've generated and used in the assemblies and map them back to the contigs to look at coverage as a metric to how well these assemblies worked, right? So if we have an assembly and it has an output and we think it did pretty well, we can map back using something like Bowtie our reads onto these, our contigs and ask, well, how much actually went into our assemblies? Sometimes this is a really low number. Sometimes it could be less than 10%. And you can be like, wow, that's not great. And that's when you might want to go back and reassess, are there some parameters I can change? Are there things that I can look at that might make our assembly a little bit stronger and a little bit better? So I'm using Bowtie as an example, but there are lots of other aligners that will take short reads and map them back to a reference sequence. This takes a while to run. So I haven't, we're not going to do it today. It also generates a fairly large output file. So we're not going to be working necessarily hands-on with that output file, but this is an example. So I took the information from the SPADES output for this artificial metagenome, right? What you have to do is you have to first index your reads or your input. So in this case, we're using Bowtie to build an index, and then we want to perform an alignment and look to see what high quality results we can look for against our contigs, right? So if you were to follow along here, Bowtie then accepts in your forward reads and your reverse reads. You use your index that you just generated to then create an output in a SAM format. So that's information. It keeps information about this format, takes the information of the alignment and the reference, and it keeps all that together and has information about where it lands on the reference and how well it lands and and how well it maps back. And so we can keep moving forward. And so if we have a SAM file, that's great. It can be generally large and cumbersome to work with. So we want to convert that information into something that we can actually work with going forward. And so one of the easiest ways to do that might be to simplify it using, calculating something like reads per base pair, right? And so in this case, we're just asking how many reads recruit back to a contig, and then we can convert them all into one standard unit and have some comparison. So that way we can look at how well certain contigs did compared to each other. Reads per base pair actually can mean lots of things to different people. So you have to be careful about the definition here. So there's one version of it where you just count how many reads recruit back to a specific contig and you get a raw count per base pair. The other things that you can do, you can actually use programs to calculate what your average coverage is for each base pair in your reference, right? So you can map it back, and you get this kind of like nice little curve that happens along a fragment of DNA. That's all kind of stochastically based off of the amplification process and the sequencing process, and you can get values back that are like, so when somebody says 7x coverage, this is an average value of how often each individual base pair is covered by your reads and at what depth it happens at, right? So a higher number here means you have more reads recruiting back to your reference, and a smaller number means you have less information, right? So 100x coverage, there's a lot of information going into your assembly. 2x coverage means that you didn't really have that much information going into there, right? It was only a handful of base pairs that did that. But we're going to calculate the more raw version of reads per base pair. And so we're going to do that using a program called feature counts, sorry. And we have to do a little bit of pre-processing for this. So we have to create an artificial file first before we can do that. And so we're going to do that using this Python script called FASTA to SFA.py. And so we're going to input, nope, no, that's the better version of it, Python. And then we're going to call it SAF. And we'll take a look about what this file looks like. And so this is just a relic of the fact that feature counts requires this type of file. All it is it takes your input information about your contigs, and it stores how long it is. And so this works really well with eukaryotic genomes, where you can actually have features that you're interested in, like chromosomal sites and regions within a chromosome that you're interested in mapping back to and counting specifically. But because we're just interested in counting the reads going back to the contigs, we're going to just treat each contig as its own entity. So if we hit enter, there's no module named bio. This is going to be a huge problem for the rest of the day, guys. So good news is the next step we can't do anyway, because it takes a long time to run. So we're going to figure this out. At our next break, I want to make sure this isn't going to derail us going too much forward. And we're going to curse Chime 2 until the end of days if we have to. This probably next step will also fail because of this, but we're going to steam ahead anyway and hope that we can solve this problem. So once we have a output from features count, which we did in this step right here, where we took our SAM file, we took our SAF file, which had information about the length of our contigs, we mapped it back. We ended up with an output that gives us just read count information. So let's see. So if we look at IDBA here, there's one that's called min 1000 read count. And so this is what the output of that looks like. And so this is just, so these are its names repeated, how long the fragment is. So the start is at the first base pair and the end is at the full base pair. These are all in the same orientation. So we have an idea of the length. And then this is the number of reads that map back to that particular contig, right? So in this particular instance, you have a million base pair fragment that then has almost 100,000 reads that map back to it. So that's pretty good. And if you could visualize this, which we're not going to do because it takes a little bit of effort to do that, and you could see that this probably will cover pretty deeply that one fragment of information. And so the next program that we're going to run, we'll take that information and just do the very simple calculation of dividing that by the number of base pairs. And then if we were doing this with genomes, we could do that for the whole length of the genome and get some information on that. But we could have followed this along. This might fail too, so bear with us. I think we want to do this with IDBA, actually. Ignore the presentation for right now. We're going to use SED from yesterday. We're going to generate a file that contains just the IDs. And so like yesterday, we want to make sure that we're providing, we don't want spades, IDBA, sorry. We want to provide a file that just has very clean IDs right here for the next step. So if we hit enter, that will work. And then we can run this Python script called convert read count to coverage. We can use this IDBA IDs file. We can output an IDBA SRR 606.249 min 1000 dot coverage. After the 1000, min 100. So if we hit enter here, I'm gonna come after Chime 2 so hard. It's gonna be a real problem here. Okay, so if there was a module called NumPy, we would be able to do this calculation pretty easily. It would generate an output that I can show you an example of in a little bit. I'll pull it up from one of the other examples that I have. And this would generate, so this just takes that information from features count and takes the number of reads aligning to the config and converts it into a reads per base pair format. And so despite having done all that with you right now, there actually is an alternative to do this that we've been working on in our lab to simplify it a little bit. And so you kind of start the same way. We're going to use Bowtie to align our sequences back to our contigs. Then we're going to convert our SAM file to a BAM file. And so the difference between this is just how much space it takes up. So BAM files are compressed versions of SAM files. So we can go through this process of compressing SAM files into BAM files. And then we can use tools within our, and mostly Elena's, program Binsanity to actually do exactly what I just kind of laid out in front of you, but in one step. Right, so the Binsanity profile step actually takes all of those inputs that we worked with individually, and we'll do that calculation for you and output a coverage file with a bunch of different ways of normalizing it if you're interested in doing that. We'll talk a lot more about Binsanity in a couple of the other sessions, so I'm not going to go too much more in depth about it, but we have a GitHub page, and it allows you to follow pretty easily along that process. So if you're interested in not following something that isn't working in the virtual machine, we can provide you with a setting that, an element that will actually work for us, all of us going forward. Okay, so we've done all this stuff. We have generated our assemblies. We've gotten some information with Quast about how well it maps back to our reference genomes. We've asked how many proteins did the output from these assemblies generate, and we've mapped it back and asked the question of how well our metagenome was mapping back to our originals. And so at the end of the day, when we compare megahit to metaspades to IDBA-UD, this is kind of the information that we get out. The number of contigs are fairly equivalent, but megahit wins in that instance. The total length of information in the contigs, so like how long it actually is won by metaspades, right? So it's about, there's a 1 megabase pair larger in data in metaspades compared to the other ones. This directly results in a slightly larger N50. And so an N50 is a calculation of if you were to take all of your contigs and lay them out, at what length is the middle point. So at what point is half of your data in contigs bigger than that? And at one point is contigs, half of your data in contigs smaller than that. So in this instance, we're saying that at the middle point for metaspades, so at 70,000 base pairs in length, we have this point where half of the data is in contigs bigger than that and half of the data is in contigs smaller than that. So that bodes pretty well that there's more longer contigs with more information in metaspades. We can ask how many predicted genes there are. And so in this case, metaspades is not the best, right? And we actually see that megahit does a better job of generating assemblies that have meaningful proteins that can be predicted there. Metaspades recruits the most reads. And then this is one that I kind of get into with people about what this actually means. So one of the advantages that megahit has over metaspades is the number of misassemblies, right? And so because we have, we're comparing to this reference set that we already know the results from, we can see that the output here, the number of misassemblies is actually lower in megahit than it is metaspades, and it's substantially lower. So about half half of the number of base pairs that have been misassembled in megahit than in metaspades. But then we ask if the assemblies map back, how well do they map back to the reference genomes? You actually see that all 3 assemblers do that fairly well within a percentage point of each other. And so the question is, which assembler is better? And I say, it really is up to you and what you're looking for. So, we're at this point in time where these are lots of alternatives to different, lots of different choices in the assembly method. But they don't necessarily always, you don't always have to pick the same one, and the same one doesn't always work the same for all types of data. And so so this will be an interesting, thing that you want to play around with. So these are 3 options. There's a couple more that are out there. These are the big players, I would argue, in terms of assembly at this point in time for Illumina sequences. And it really is one of those things that, depending on what your goal is, you might choose a different assembler over one or the others. And so that's what I just said there. It depends on your project, and it depends on your sample and your environment. Okay, so now we have assemblies. Yeah.
Is there an acceptable number for N50 or percentage that you can say assemblies are good enough?
Is there an acceptable number for N50 to say if our assembly is good enough? The answer is no. It will depend on your sample, and it really depends on what you're aiming for, right? And so if you have a complex system and you are not sequencing 100 million base pairs or something like that, you might expect your N50 to be low. But if you're working with a simple system and you've done, 1 billion base pairs, you would expect it to be high. And so all you can kind of do is in your brain, once you know what kind of to expect, is to assess the output that you're getting, right? So if you have a simple system and you have a really low N50, there might be a reason for that you need to explore. And if you have a really complex system and you have a really high N50, well, that's great. Yeah, percent recruiting is the same thing. It all comes down to how complex your system is. And so there's no one right answer to this question. And that's why it can be frustrating and difficult to do. And people like to go through this as fast as possible. Because you don't want to spend a lot of time thinking about, but if I make this one little change, maybe things are going to be really fundamentally different for me. But yeah, it really just depends on your system, which is a awful answer to hear, but it unfortunately is the truth.
Can you give a range for a really low complexity system like acid mine drainage?
Yeah, so I would think in a really low complexity system like acid mine drainage, you probably would be expecting an N50 that's probably over 100 KB at that point, right? So 100,000 base pairs, because it is simple, and you should be able to, get your assembler. Your assembler should be able to parse that pretty effectively. Sediments, if you get 700 base pairs, you're probably in a good environment right there. Yeah. But there's no hard, fast rule for that, right? And so you just have to, that's why you're looking at all these other metrics to assess how well you're doing. That's why you're doing percent recruitment. That's why you're looking at how many genes can be predicted, because there's not one hard, fast answer for that. So what are we gonna do next for the rest of the day? We're gonna do some binning, some annotations, some phylogenetics, and look at and try and assess how well we can do the rest of this. With Chime ruining everything, but we'll solve that in the break. And so we'll take a break right now. We'll come back in 10 minutes after everybody's had a chance to refresh, and we'll move on to the next part, which will be binning.
Should we use mother?
Oh, shit.
I don't think it's that